All right, we're going to talk about DNA replication steps. There are 3 major steps with DNA replication. The first step is going to be initiation. Replication always occurs at a point of origin. That way it has somewhere to start. The second step is elongation. Enzymes are going to be forming new DNA strands because we have a semi-conservative model. They're synthesized by an enzyme DNA polymerase. Now, there are different types of DNA polymerase. This is an enzyme that forms new strands of DNA. We have DNA polymerase I, and we're going to talk about DNA polymerase III. The third thing is it has to finish somewhere, so it has to have a termination. It has to stop. Now, if we were talking about DNA termination, basically, they're going to finish differently. So replication ends. When we say terminated, it ends. But it is going to be terminated differently in prokaryotics versus eukaryotics. Now, as far as prokaryotics go, I'm not going to discuss a lot about prokaryotics, basically because you're not going to have to learn the great details. This is going to end at some point, but really what you need to understand is all the enzymes that happen in DNA replication. So I am going to draw out DNA, and I'm going to take all the enzymes and I'm going to basically go step by step by step with the replication fork and describe what's going on with the enzymes. I need a whole picture for this because it gets confusing. So I'm going to take it a little bit at a time. Now, we know that the first thing that has to happen is you have to take your double helix and you have to create the replication fork. You have to unzip it. Now, when you talk about unzipping DNA, it always occurs by the enzyme helicase. Helicase is always going to unzip or unwind DNA for replication. That's the first step that has to happen. Now we're going to have other players involved, and this is going to get really detailed. So, first thing is this is going to be DNA replication. And the enzymes involved. All right, so we have our replication fork. The first enzyme that we're going to draw right here is going to be helicase, which I've already discussed. Helicase is responsible for unwinding DNA to create the replication fork. Now, during replication, we have to prevent DNA from rewinding because it's really tightly coiled. So there's proteins that are going to be found really close to helicase that are responsible for keeping DNA unwound so that replication can occur. Now, these are proteins that literally attach to the sugar phosphate backbones that are going to anchor the sugar phosphate backbone, so they bind to it and they prevent DNA from rewinding. SSBP. SSBP is called single-stranded binding protein. So, single-stranded binding protein. Single-stranded binding protein is going to prevent the replication fork from rewinding. We shorthand it as SSBP. So, I'm going to actually do 2 papers because it's going to run out of time or room. Helicase unzips or unwinds DNA for DNA replication. SSBP, single-stranded binding protein, prevents DNA from rewinding during replication. Pretty important. Now, we have to understand that DNA is anti-parallel because everything is determined by it being anti-parallel. So, this end is going to be the 3' end, and this is going to be the 5' end. On the opposite side, the 3' end with the 5' end. Now, we said that the primary enzyme that's used for DNA replication is DNA polymerase. So, DNA polymerase, there's several of them. So we're going to say that this is DNA polymerase III. DNA poly 3. This is the easiest way to draw it. Now, DNA polymerase III is the primary enzyme that does DNA replication. Now, when we talk about DNA polymerase III, DNA polymerase III is going to build new DNA from 5' to 3'. So it has to start at 5. So DNA polymerase III on this left side is going to build a new DNA strand that goes in the same direction as helicase, but it only can build it from 5' to 3'. Now, when we talk about DNA replication, this is easy because helicase is moving in the same direction. So we talk about this as being the template strand. But this side always happens first. So the template strand of DNA that goes in the same direction from DNA polymerase is going to be called the leading strand. The leading strand is the DNA strand that is synthesized continuously. It's always going to be on this left side. The leading strand is the strand of DNA synthesized continuously. So DNA polymerase III on the left side is working from 5' to 3'. It's really easy for it to build complementary DNA on this side. Now these are going to be complementary nucleotides complementary to the template strand. So the left side works in the same direction and it's called the leading strand. Now the opposite side is where it gets tricky. A new enzyme has to get the opposite side ready because it doesn't move in the same direction as helicase. So we have an enzyme which we call RNA Primase. It's also called RNA Polymerase. Now, RNA Primase is going to prime the opposite side so that DNA polymerase can know where it needs to go and begin. So it's like, it's not working right because it doesn't know where to start because it's not going in the same direction. So what you do is you go put a primer down. So it's no different than a primer that would be something that you use as paint. So the enzyme RNA Primase, also called RNA Polymerase, on the opposite side is going to prime basically the opposite side so that DNA polymerase knows where to begin DNA replication. Now it's going to prime it in several places. So it is not made out of DNA, it's made out of RNA. So it primes it in several different areas. So this comes down and it primes it here, it primes it here. It primes it in several different places. So RNA Primase. RNA Primase is also known as RNA Polymerase. It is going to prime the lagging strand of DNA so DNA polymerase knows where to begin DNA replication on the opposite side. You always do the left side first. My pen is, or my marker is going bad. RNA Primase is also called RNA polymerase. It is going to prime. Prime means to get ready. The lagging strand, which we're about to label, of DNA. So, DNA Polymerase III knows where to begin DNA replication on the opposite side. So now we're working on the right side. It's priming it, it's getting it ready, but it is made out of RNA. Primase means to get ready. It is also called RNA polymerase. Now that RNA polymerase or RNA primase has primed the opposite side, your DNA polymerase is going to come to the opposite side and it's going to start to build DNA in the opposite sections. Now, there's a problem with this. First of all, this is lagging. It was slow. So the DNA polymerase came up, but also this is going in the opposite direction. So we call this the lagging strand. The lagging strand is the strand of DNA that is synthesized discontinuously, meaning it's fragmented, and it creates Okazaki Fragments, named after the scientist that discovered it. So the Okazaki Fragments are the pieces of DNA. So this is DNA, and this is RNA, and this is DNA, and this is RNA. So it's lagging strand because it's going behind, or it's slow. It's on the right side. But the fragments are pieces of DNA and RNA, but that's not good. We have to replace that. So the leading strand is easy because it works in the same direction as helicase. So literally, when we do the steps of DNA replication, let's go back. You first have to unzip it. Helicase is always going to be the first step. He unzips DNA. Your single-stranded binding protein on either side is going to prevent DNA from rewinding. You always go to the leading strand first. So, number 3, step number 3 is you're going to create the leading strand by DNA polymerase because it works in the same direction. On the opposite side, RNA polymerase is going to prime or get the lagging strand ready because it's slow. And in the process, DNA polymerase knows where to start on the opposite side. And what we do is we create Okazaki fragments, which are fragments of DNA and RNA. So we're obviously not done with DNA replication because by the time this is done, it has to be completely DNA, not DNA and RNA. So now I need to add a new picture because we've got to get rid of all this DNA. So we have our list here, and we're going to come back to it. So, DNA replication continued. You draw your replication fork. You always label your 3' and 5'. It's anti-parallel. We unzipped it through the enzyme Helicase. We held it apart with single-stranded binding protein, so SSBP on either side. The side that works on the same direction as Helicase is going to use DNA Polymerase III, so we can write D poly 3, the circle. This is going to create your leading strand. It leads. Now, the next step that you have to do is you have to get rid of your RNA. So, we're going to use an enzyme. Trying to find a good color. An enzyme called DNA Polymerase I. Number 6. DNA Poly 1. His job is to remove the RNA on the lagging strand. He literally is going to go and replace all the RNA with DNA. So we can write this on our list. DNA Polymerase, it's an enzyme, 1. Enzyme that removes RNA on the lagging strand and replaces it with DNA. RNA polymerase. So what happens on the lagging strand is now you have DNA from DNA polymerase I that replaces the RNA, and you still have DNA polymerase III so now you have 2 enzymes. DNA polymerase III did the original DNA, and DNA polymerase I replaced all the RNA. So you have DNA from DNA polymerase I, you have DNA from polymerase III, you still have DNA from polymerase I, and DNA. Now, you still have Okazaki fragments. Okazaki fragments are only found on the lagging strand. Discontinuous creates Okazaki fragments, but now your Okazaki fragments are DNA, 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 and DNA, which is good. So we've replaced number 6, all the DNA or RNA that needed to be replaced. And the last thing that we need to do is link all these Okazaki fragments together. And that's where the last enzyme comes in. Number 7 is ligase. Ligase is going to link the Okazaki fragments on the lagging strand. Now I get that that's in yellow. I can go through and write that. Number 7, step number 7. The enzyme ligase is going to link the Okazaki fragments on the lagging strand. That is step-by-step replication and the enzymes involved. So if you're doing a list of enzymes, you would need to add ligase. Enzyme, we know it's an enzyme because it ends in ase. That links Okazaki fragments on the lagging strand. And now you have a list of enzymes and what they do. Go through this several times until you understand it. So really, the last step that would occur is once you link all the DNA together, it would really split into our semi-conservative model, and we'd have one old strand and one new strand. So that's DNA replication and all the enzymes. Now, prokaryotic replication and eukaryotic replication is a little bit different, but I think this is enough to help you understand the enzymes.